Hi, this is Dr. Ricardo of the Department of Anatomy and Cell Biology, School of Medical Sciences. I present to you the third week, which involves the formation of the trilaminar disc, and this is described as gastrulation. This is a sequel to the earlier video on week 1 and week 2 updated. So the week 2, as we covered earlier on, we said gives two structures, as in a pair of them. So let's quickly go through that. So trophoblasts will form cytotrophoblasts and syncytiotrophoblasts. Embryoblasts will be divided into epiblasts and hypoblasts. And then the extraembryonic mesoderm will be divided into splanchnopleuric and somatopleuric by the xylem, or the species that coalesced. Now, the two cavities at this stage will be the amniotic cavity and then the yolk sac. So the amniotic cavity will be above the epiblast with its membrane. So that's the cut edge of the amnion. So if it is extended all the way for it to touch the edges here, there will be a space within it. That is a space above the blue region, which is the epiblast. That will be the amniotic cavity. And then the yolk sac will be the one below the yellow, that's the hypoblast. So that is the wall of the yolk sac if it is extended. All right. So remember, you have the primary yolk sac and then you have the secondary yolk sac. So that is what we have to bear in mind. In gastrulation, the formative process by which the three germ layers are acquired will ensue. And the precursors of all embryonic tissues come from these three germ layers. And there's also the axial orientation of the embryo, i.e. the head region and tail region will be established. During gastrulation, the bilaminar embryonic disc is converted into a trilaminar embryonic disc made up of the three germ layers, ectoderm, mesoderm, and then endoderm. So if you look at this, the space here will be the amniotic cavity, and therefore that will be the epiblast. On top of the epiblast, you have a primitive streak and then a primitive node. Within the node is a pit. So there is going to be invagination of cells. Cells are going to migrate through the primitive streak, okay, through the pit, and then they fall down on top of the hypoblast. So you can see this green particle and some red particles as well falling in between the epiblast and the hypoblast. So in a larger or magnified image, this is what you see. So you can see the progression, the movement of the cells, epiblastic cells through the primitive streak and then the pit. So eventually, some of them are going to displace this hypoblast and become endoderm, those that will be left suspended in between the hypoblast, the former hypoblast, and then the epiblast will become mesoderm, and the rest of the cells that are left within the epiblast become ectoderm. All right. And then also, there are a lot of interactions that go on while these epiblastic cells are migrating through the primitive streak. Okay, so several genes are turned on while they pass through the primitive streak. All right, and they are modified. So they get some modification as they move along. And then per the position they will find themselves, they will either become endoderm or mesoderm or ectoderm. And then those that will fall in the midline will form the notochord. So again, primitive streak, so that means that that's the surface of the epiblast, okay? And these are the cells that are migrating through the streak and falling into the pit and therefore onto the hypoblast, which is green in this color. So the yellow ones are the epiblastic cells. So they are filling up the space between the hypoblast and then the epiblast, which will therefore become mesoderm. Those that will displace the hypoblast will become endoderm, and those that are left in the epiblast will become the ectoderm. So we also said that during gastrulation, the orientation of the embryo is established, which means that you get a head region and then a tail region. So usually the head region is a bit larger than the tail region. All right. And then also the primitive streak is usually toward the tail part. That's the left and right. And then that will be where the buccopharyngeal membrane or oropharyngeal membrane will be located. Right. So that will be the axial orientation that will be assumed by the embryo. So when we cut this portion of the embryo through the primitive streak here, and then we look at the surface where the cut was done, this is what you are going to see. So see the primitive streak, and that is the node. So that's primitive node. And these are epiblastic cells moving. So some are displacing the hypoblast to become endoderm, right? So that is what goes on. Eventually, it is still filling up, and then the filling up there will become mesoderm. So another diagram just depicting the same thing, primitive node, primitive streak. Then you have the epiblast, then hypoblast, the invaginating cells from the epiblast going to form the mesoderm. Gastrulation is the beginning of morphogenesis or development of body form. So the extensive cell shape changes, rearrangement, movement, and changes in adhesive properties contributing to the process of gastrulation. So cell movement and shape changes during human gastrulation. As you can see, the epiblast and the cells that are moving in, once they get through the primitive streak and are falling through the pit, as I said earlier on, they have some interaction with the primitive streak, and so there are changes and modifications. So you can see some of them are bottle-shaped, some of them are stellate-shaped. Okay, so their shapes will also determine their fate in future. All right. So the blue line is, or arrow is showing the direction or migration of cells in the epiblast, and the red one is migration of cells internally after leaving the epiblast. So at the end of gastrulation, what do we get? We get ectoderm, endoderm, and then mesoderm. But we call these intra-embryonic, okay, as against extra-embryonic. Remember, we had an extra-embryonic mesoderm formed in the second week. Okay, it is this extra-embryonic mesoderm that split into two to form the somatopleuric and splanchnopleuric. All right, so if you want to differentiate between those cells that were formed in week 2 as against that of week 3, we mention them by giving this intraembryonic, prefix to them. So intraembryonic mesoderm instead of just saying mesoderm. All right, so that you can differentiate it from extraembryonic mesoderm. Then that is endoderm. You can also say intraembryonic endoderm, and then intraembryonic ectoderm. But by convention, we know that because most of them do not stay to term. Whenever we say mesoderm, we know we are talking about the one that occurred in 3rd week, which will stay to term. Okay, and then ectoderm and endoderm alike. So after gastrulation, yes, that's what you find, trilaminar disc made up of ectoderm, mesoderm, and endoderm, or intraembryonic ectoderm, intraembryonic mesoderm, and then intraembryonic what? Endoderm. And that is the primitive groove. And then you can see the yolk sac. That's the secondary yolk sac. So you get secondary yolk sac by the time you have gastrulation or 3rd week. You definitely have the secondary yolk sac. The primary yolk sac would have, diminished as we saw in week 2. So again, after gastrulation, our emphasis now will be on the intraembryonic mesoderm, which will have 3 regions: the paraxial mesoderm, the intermediate mesoderm, and then the lateral plate mesoderm. That is the notochord, which falls just beneath the primitive streak or the primitive groove, okay, notochord. As we see, it can cause some problems when it persists, but in the adult, you actually find it in the vertebra, okay? So In the intervertebral disc, you have what we call the nucleus pulposus. Okay, that is the normal remnant of notochord in the adult. But there are a lot of problems that this notochord can also, present with when they stay. So let's look at formation of the neural plate and neural groove. So as you can see, you can have the caudal end and then cranial end. As I said earlier on, the cranial end should look larger than the caudal end. So you have the notochord in there, which is going to influence the ectoderm above it to thicken to form what we call a neural plate. Okay, so the notochord influences or triggers the ectoderm above it to thicken to form the neural plate. So at the end of the day, you end up having a neural plate, and then there's a groove within the neural plate. It starts forming a groove. So you can see that there's curvature here compared to this one. These ones are just straight lines. This one is curving. So there's indentation in the midline. So as we go along, we want to cut through it, plane of section through it, and then look at the half of the cut surface. So this is what we are going to see as a primitive pit. That's the notochordal process. Remember, it's a mesodermal structure, okay, because the cells fell within the epiblast and hypoblast. But because it fell right directly below the groove, it assumes a different name, notochord. All right, so embryonic ectoderm is there, and then that is the embryonic endoderm. All right, that's the cardiogenic area, septum transversum. We'll talk about it during cardio. That is allantoid. We'll talk about it during GI, and then also renal. So when we come to this point, the primitive pit is there, and you can see that there is elongation of this process, the notochordal canal, okay, going through the notochord. So that's the notochordal process. So when you look at it in this section of view, that's the notochordal canal and then notochord, and then flanking each side will be the intraembryonic mesoderm. Okay, starting with paraxia mesoderm first, then intermediate and the lateral plate mesoderm. So as we continue, you see that the notochordal canal is increasing. All right, you can see that. But there's a part here, cloacal membrane, where you don't have any intervening mesoderm, the cloacal membrane, okay? And then a similar thing will occur at the head region where we have the buccopharyngeal membrane. There will be no intervening mesoderm. It's strictly ectoderm and endoderm, combined. All right, so that's primitive pit notochordal process, and then the pit or notochordal canal, as we saw earlier on. So here too, that's the pit, and then that's the neural plate. That's the oropharyngeal membrane I mentioned not long ago that you don't have any intervening mesoderm. So there's no red, in between the blue and the yellow. It's strictly ectoderm and endoderm. So that is the notochord, and it is joined to the endoderm at a point until it forms this spherical mass. And you can also see the grooving that occurs in the neural plate. You can see that eventually this groove is going to be indented, okay? It will be deep, and then you have what we call neural fold emerging on each side. It will continue until this neural fold meet each other. All right, so you can see here that the neural folds are now meeting each other. Once they do that, the rest of the ectoderm will join, and then this will form a tube that becomes the neural tube, which will eventually form the brain and spinal cord. All right, and you have your notochord there, you have your intraembryonic mesoderm, paraxia mesoderm, intermediate mesoderm, lateral plate mesoderm. Most of the paraxia mesoderm will form somites, which will help in the musculature of the fetus. Then the lateral plate mesoderm splits into two with the space there. So you also have splanchnopleuric and then somatopleuric. The somatopleuric will go along the amniotic membrane, and then the splanchnopleuric will go along the yolk sac, okay, the secondary yolk sac. So this is a summary of what happens in neurulation. That is the neural tube formation. Okay, so neuroectoderm, there's indentation, and have neural folds. Look at the coloration, deep blue and light blue and very light blue. Then that's the notochord, which influences this indentation or groove formation, okay? Eventually, you can see that the indentation is even deeper, forming the groove, neural groove. And then the light blue is now getting closer and closer, and it's also getting closer. So you realize that a portion of the light blue, these ones, will not be part of the neural tube. And it's the surface ectoderm. It will meet. Once it meets like that, it continues its surface ectoderm outline. Then the other light blue that did not form part of the neural tube becomes the neural crest cells, which form a lot of structures in the body, as you see, from melanocytes, to wherever you can think of. They are always everywhere in the endocrine system, digestive system, the oral cavity. They are there everywhere. So that's neural tube, developing epidermis, all right, coming from the ectoderm. Then that is the neural crest cells. Even in the nervous system, they form the spinal ganglia, in the spinal, cord area, where you have the afferent fibers entering the spinal cord. Okay, the dorsal root ganglia is actually formed from the neural crest cells, so developing spinal ganglion. So this also a flow chart showing how the process continues. So that's neural plate, neural crest, neural groove, surface ectoderm, neural crest, and this will be neural tube, neural crest, developing epidermis, and then there you go, you form your neural tube complete. So I was talking about notochords and their remnants. So persisting notochords can form chordomas, and this may form from vestigial remnants of the notochordal tissue. Approximately 1/3 of chordomas occur at the base of the cranium and extend to the nasopharynx. Chordomas also grow slowly, and malignant forms infiltrate bones. So this is where you find them at the base there. Okay, in between, this is the pons. All right. And then that's the midbrain. So it is forming around the base there. And there's another diagram or picture showing the position of the chordomas, that's remnant of notochord. Then also when the primitive streak does not degenerate and also persist, it can form teratomas. So when the teratomas occur at the coccyx area or the tail region, it becomes sacrococcygeal teratoma. And when it occurs at the head region, it becomes the oropharyngeal teratomas. Okay, so that's another sacrococcygeal teratoma. Read on Hirschsprung disease and neural tube defects, okay, NTDs. Thank you very much. I hope this video helps.